Hello, everybody, and welcome to kitchen table number 42. So it is my great pleasure to introduce our first back-to-back dual speaker event. I'm Diana Mitre, I'm your host, and I'm joining you from Dewpoint's kitchen table in Boston, Massachusetts. So before we get into today's talk, a few housekeeping messages. This talk, as you know, is a live event and will be recorded. So feel free to turn on your camera and follow along with the speaker, but please no on-camera shenanigans. We would prefer to keep the questions to the built-in Q&A breaks. So feel free to type your question in the chat at any time. And during the Q&A, I will call your name, ask for you to unmute your microphone, and at that point, you can ask your question directly. If for whatever reason you're unable to talk, please let me know in the chat, and I'll ask the question for you. The event recording will be posted on condensates.com within a week. So now let's go to the part for which you're here. It is my great pleasure to introduce today's first speaker, Dr. Agnes Toth-Petroczy. She's a group leader at Max Planck Institute of Molecular Cell Biology and Genetics in Dresden, Germany. Agnes is renowned for her contributions in computational biology and protein evolution. She has earned a master's degree in chemistry from Eötvös Loránd University in Hungary and a PhD in life sciences from Weizmann Institute of Science in Israel. She completed her postdoctoral training right here in Massachusetts at Harvard Medical School in Brigham and Women's Hospital. Throughout her career, Agnes contributed to our understanding of evolution of disordered proteins and their contributions to Mendelian diseases. She mentored numerous trainees and supported equality, sustainability, and community outreach efforts. In 2025, she was honored with the Ernst Schiering and Embo Young Investigator Awards. Since 2018, as a group leader, she contributed to the development of several bioinformatics tools that facilitate the study and advancement of intrinsically disordered proteins and biomolecular condensate science. One of these, CD-Code, is the topic of today's talk. So without further ado, Agnes, the floor is yours.
Thank you, Diana, for this very nice introduction. And also, thank you for giving me the opportunity to present CD-Code. I still remember when we started brainstorming about the idea of creating a knowledge platform about condensates back in 2020 during the COVID lockdowns. And back then, there wasn't even this AI revolution at sight. And now we know basically that AI is revolutionizing our lives and also drug discovery. So in my lab, particularly, we are interested in intrinsically disordered protein regions, or IDRs for short. And these proteins and regions don't have a well-defined structure. And because of this, they are also considered undruggable. And often these disordered regions actually drive the formation of biomolecular condensates, which could be a way around this and targeting actually with these condensates instead of the disordered regions. And while AlphaFold, as one of the breakthroughs in structural biology, basically solved the problem of sequence-to-structure paradigm for ordered proteins, and it can predict the structure of the ordered regions, it still cannot predict the structure of the disordered parts of the proteins. It gives only low-confidence predictions, and this is something my lab is interested in. How can we actually advance the sequence-to-function paradigm of disordered proteins? But let's discuss, why does AlphaFold work, or what were the requirements for it to be developed? And of course, the developments in computer science and the breakthroughs in new AI algorithms contributed, as well as the hardware developments of the recent years. But very importantly, also the training data was absolutely necessary to develop this tool. And here we are talking about two types of data in case of AlphaFold. So it needs evolutionary data, so all the sequence information from hundreds and thousands of different organisms that we have sequenced in recent years, and the decades of structural biology efforts of actually collecting structures in PDB as the main resource. So the community basically agreed to deposit these structures that they have solved in the PDB and also develop standards for what is a good structure. And if we just have a quick estimate of what is the cost of data actually in PDB that I asked ChatGPT, it comes up with this 3 to 20 billion euro estimate for actually the data that was deposited there. So, but I would argue there is even one more ingredient that is important for success and for these breakthroughs, which is the challenges that the communities actually develop. And this is, for example, the critical assessment of structure prediction, or CASP, that AlphaFold won in 2021, is when the community actually agrees what are the questions that we want to tackle, what are the challenges to solve, and what are the metrics of success. And there are several other challenges for predicting interactions or functions, also predicting intrinsic disorder. There is no challenge yet for condensates. And all of these, of course, use the data sets and databases that were developed and can be used for training and also for evaluation, for structure, sequences, functional annotations, and also disorder. And now, as of 23, when we released the first version of CD code, there is data also on condensates that followed 4 previous databases that were mainly focusing on liquid-liquid phase separation, and also collected proteins that are involved in those. So our goal was that we would like to assemble the condensate data and kind of catalog it in a way that it's useful for systematic discovery and also for AI modeling. And since the condensate field is very new, there is actually a zoo of different terms that you can find in the literature, and what the community actually defines as a condensate and what experimental evidence we actually accept as a proof for a condensate to exist. So we decided we will go with the term biomolecular condensates. This was coined in 2017 in a landmark review. And we distinguish basically biomolecular condensates from synthetic condensates, which would be condensates or phase separation actually assayed in vitro when only one or few components are actually studied, from condensates being observed in cells or in the organisms themselves. And regarding the components, the protein components, we distinguish drivers, which would be proteins that are essential for the formation of the condensates. And if we would knock out this protein, we wouldn't actually see them. And often these are like scaffolds and kind of binding many different components. While members, on the other hand, or clients, they can partition in and out of the condensate without compromising its integrity, and they could potentially modify its function. Because of this, all these many terms, right, we had to already make some decisions. So we decided we will build in a crowdsourcing functionality into the database so the community can contribute and edit it. And we named it Crowdsourcing Condensate Database and Encyclopedia, or CD code for short. And this was truly a collaborative effort between Dewpoint, where from the beginning, Diana was our main contact and the main lead, and Tony Hyman's group and my group, the first version headed by Nadia and Deep. And the first version already had around 10,000 proteins and 200 condensates, which since then actually doubled. And in the background, there are many other people contributing, of course, both on the software side and on the data side. And we even organized these community events at my institute at MPI-CBG, which we call condensathons, when we get together, have free pizza or celebrate with a cake and discuss papers and decide on the manual curation of many of the entries. Because although we can use AI, and we of course use LLMs to help with the curation of the papers and the data, there is need still for human checks and interactions. So our policy was that the curators actually become co-authors. So if you are interested curating data, then join us and you can be part of the next release and potentially the next publication. So the second version of CD code, so while in the first one we focus mainly on the function and the components, in the second version we decided also to include data on biomedical relevance, that has biomedical relevance. So we started curating infectious condensates, that are condensates that form upon viral or bacterial infection, and drugs that impact condensates that are called CMODs, and condensatopathies, which are diseases that are linked to condensate dysregulation or malfunction. And this second version was headed in my group by Xenia and Maxim. We are also putting effort into improving the user experience, and you can see many people actually just browse on the website, and these are the terms that most people, that are kind of the most commonly searched terms on the website, like highlighting the most famous phase separating proteins. But also we have access with, for programmatic access, we have an API that we have now improved documentation for mainly computational research or AI developers as well. So just to give you an example here, you can see and select, for example, infectious condensates. That gives you a long list with the different components annotated, and then a description, links to different other resources cross-referenced, and then always gives you basically an evidence for what type of experiments are supporting, let's say, that this protein is part of that condensate. And this is actually crucial because depending on what type of evidence you're looking at, for example, many times we just look at microscopy-based condensate kind of identification and localization, and we actually don't know what other proteins are in this condensate. So if you just look at the distribution of how many proteins a condensate contains, many of them looks like they have only a few. But a few condensates have been actually purified, and mass spectrometry analysis suggests that there are hundreds and even thousands of different members inside these condensates. Also, if we look at the proteomes of different condensates, we can see that they overlap. And for example, in stress granules and p-bodies would share 300 different members that are shared between them, also potentially suggesting their different origin. So many proteins actually can partition into several different condensates. We also looked at the disorder content, of course, and we can clearly see if we compare to the human proteome, the different length distribution of IDR regions to the condensate proteome in CD code, that there is an enrichment for IDRs. However, there is still 21% of proteins that have no disorder content at all, and they are still found in condensates, which could be potentially modulators like kinases or, let's say, clients. But at the end, it turns out that even drivers can be fully structured. So we can put them in this continuum where there are disordered proteins driving condensates like FAS, but there are also fully structured proteins like SPOP that are found in nuclear speckle, driving nuclear speckle formation. So this data that we have now systematically in CD code enables testing certain hypotheses that the field had, or also highlights the unknowns, let's say the members that we still don't know. And it also enables machine learning and AI applications. So in my lab as a computational biologist, we're also interested in developing methods for condensates. And particularly, we are interested in predicting from a sequence of a protein if it would form condensates and which particular condensates it would localize into, and also how would mutations in the protein sequence impact the condensate. So I will briefly introduce you our method, that is PICNIC, that predicts condensate formation, but also CD code data already kind of was used to develop ProGPS, which is a tool that can predict the specific localization of a protein into 11 different condensates developed by Rick Young's group, and CatGranule2 that is predicting the impacts of mutations on the protein sequence. So it's great that we have CD code because this can be used as kind of positive examples for a machine learning predictor. But for supervised learning, we also need negative examples, which is much more tricky to find. So Anna in my group thought about basically using the protein-protein interaction network in human in this case, and define as negative those proteins that are not known to interact with already existing condensate proteins that we have already proved that they form condensates. So they're at least 2 steps away in this interaction network from known condensate proteins. And this was our way of having kind of an unbiased negative data set for training a predictor. And our predictor is called PICNIC, that stands for proteins involved in condensates in cells. It uses sequence and structure-based features, and then at the end basically classifies proteins into yes or no forming condensates, regardless of also structural disorder. It predicts around 1,500 new condensate proteins in human that have not been characterized yet. And we tested actually some of them experimentally with Harry, Tony Hyman's postdoc. And we were happy to see that overall it has an 80% accuracy, as we can see here in blue, all the proteins that we tested and indeed form condensates. And this can have indeed a wide range of structural content, can be highly disordered, but also highly structured. PICNIC can also be used, of course, not only in human data, but across any organism or even any synthetic sequence. So we wanted to see what are the condensate components of different organisms. And while we know that disorder content increases in evolution, so compared to bacteria and archaea, there is a lot more disorder in eukaryotes. There was an expansion. We don't know the same about condensates. So when we looked across 24 different organisms across the tree of life, we were surprised to see no correlation with disorder content. It seems to be that condensates are ubiquitous. And in archaea, there is even a higher condensate content predicted than, let's say, in mammals. So if condensates seems to be then essential and ubiquitous across species, then also their dysregulation may lead to disease. So this is what we started now collecting. So basically led by Alicia and Diana at Dewpoint, we now have a list of different diseases that are known to be associated with some condensate dysregulation. So basically we collect which condensate is impacted in what disease and what dysregulation type that actually links to. And this can be either a direct link to the pathophysiology of the disease, or these misregulations could be treated as a biomarker of the disease, a kind of marking what's the disease. So when we look at most condensate-related diseases are actually either infectious or cancer or neuronal. And the most likely condensate to be affected is stress granules, but there is also a long list of other condensates that have been already implicated in diseases. So if we can observe that under disease conditions there are differences in condensates, then there could be potentially a way to target these condensates. And this is what these condensate modulator drugs are doing. So we, according to, again, Dewpoint's landmark review, we classify them into these 4 categories. So a CMOD could dissolve a condensate or induce the formation of the condensate, changes localization, or changes properties like material properties or shapes, which we call morphers. So we collect now all these small molecules in the database and cross-reference them again with known other chemical databases. And when we looked at these over 350 CMODs that we right now have in the database, then interestingly, most of them either dissolve or induce the condensates, but some actually can have different effects depending on which condensates they impact. And also they have different modes of action. And when Diego from Dewpoint looked at the chemical space of these different molecules, then he couldn't see any clustering based on the mode of action. So there is a wide variety of different chemistries that can give rise to the different actions depending on which condensate we are also looking at. So what we are thinking about the future is that so far we collected information on condensates in CD code about is there a condensate or not? And that was kind of the evidence that we were looking for. But we should also think about describing these condensate phenotypes and also a bit more quantitatively, actually by looking at the images and classifying them into these different categories. And that's what we started working on now. So developing a tool that can take hundreds or thousands of images and actually describe the condensate morphology, spatial distributions, or phase behaviors, let's say concentration dependence, and give us a way to actually quantify the different condensate phenotypes. So looking forward in the next version of CD code that we call ICD code, we'll collect also these phenotypes and the images that are actually the raw data, as to say, to why we think these condensates and these different proteins are actually there. And we also would like to collect information on mutations in the protein sequence that would impact the condensate behaviors. And if you have any other ideas what we should include and what type of developments we should do, what's the new data we should collect, then please reach out and write to us. And with that, I would like to thank you for your attention and happy to take questions.
Thank you, Agnes. Do we have any questions online? Please raise your hand or type your question in the chat. Oh, right, we have a question here from Italo.
Hi, how is context dependency included in the database and also in the tools that your lab have been developing?
You mean context dependency would be the different cell type or?
Yeah, like the condensate formation, which context, tissue type, and even condition, for example.
Yeah, so at the moment we don't have it actually. We started collecting this information, but it's not yet visible from the front end. We started collecting the cell types. So this is a very good direction forward. And I don't know what other context we should record. So please suggest what you think we should add as an information, because for now we basically have only the experimental evidence, and this, if it's in cells or in vivo or in vitro, we could add the cell type. So what else should we collect?
Yeah, I think cell type is a good start, but also the experimental evidence, the type of experimental evidence, because of the biases of each experimental approach, right, to detect them. Yeah, these are the two things.
So the experimental evidence we have, like roughly, we have if it's microscopy or FRAP has been done or mass spec. And the cell type we can add, and I think if we have the images, so if we actually start collecting the raw images, which would be actually great if the community would also think this is useful. So similarly how PDB collects the 3D coordinates, right? This is basically what you have to deposit. If you would deposit actually the raw images, then from that it's very easy to classify what was the cell type, what were the conditions, what was the concentration, because that's something that we could actually also measure from these images in a standardized way. Maybe it's even easier than digging it out from also publications.
Thank you. Giancarlo, would you like to ask your question?
Yes, I don't know if you can hear me.
Yes.
Very nice talk, thank you. And I'm just curious to know if your database can provide information about the structural changes of the proteins within the biomolecular condensates when we are referring to structured proteins.
No, not at this point, no. So it's basically only presence, absence of a given protein in a condensate. And it's also kind of the union of all potential experiments that have been done about this protein. You would have all the links to the corresponding publications, but we don't actually record the conformations. And I think there's also not that much data yet in the literature about this, but it would be actually great to have that information.
Indeed, that would be definitely something very important to add to your database. Maybe, well, we can think about it. Yes, good to know. Thank you.
We have another question in the room from Django.
So this looks very interesting. However, I understand it's quite complicated. So my question is that, do you have any idea or any plans to build a condensate map across different cell types or different disease and use it to guide the drug screening?
Yes, so the cell types we should add. I think that's still missing. So to have this full map of information, and I think also the condensates, how to say it, like we call them canonical condensates, would be the one that are kind of named. Let's say nucleolus and nuclear speckles, but actually there are a lot of condensates that just have a few components that are named after the proteins. And this could be actually the same condensate, just we don't know that these proteins potentially colocalize. So I think we don't even know how many condensates there are still in the cell. That's why I actually didn't say that number, because I think this will change.
Yeah, that's true. So my concern is that I don't care about single proteins, single pathways. For example, if two countries are fighting, the DoD building, they may be made up by different peoples or different information, but you know the image, then you can hit it, then you can hit your enemies. This is my question. So with that being said, if you can build a condensate map, let's say different building has different functions. Then in future, your screen drugs, then okay, for cancer, let's say breast cancer or either ovarian cancer, then okay, we can hit this building, then we can we can screen drugs. I just give you an example. I don't mean the real world. It's not as simple as this one. But however, if you have a map, certainly it's a very crude map. It may be useful. That's my key point.
Yes, maybe in Francis's talk, actually, there will be more information of this map. and
thank you.
how we can aid that. Yes.
Yeah, I think we're going in that direction with the condensatopathies recording, but it's building. It's not there yet, but I think that's on path with what Django was talking about. All right. Well, if there are no other questions, let's thank Agnes, and I will introduce our next speaker, which is Francis Carpenter, the head of data science and engineering at Dewpoint Therapeutics. And he will continue the story, telling us how tools like the ones that Agnes introduced could be utilized and integrated into the drug discovery process. Francis has a PhD in neuroscience from the University College of London, where he combined in vivo electrophysiology, chemogenetics, and computational modeling to study the mechanisms underlying the brain's representation of space and how this supports long-term memory. Prior to joining Dewpoint, Francis was an associate partner at McKinsey & Company and at Quantum Black, where he led cross-functional teams supporting biopharma clients to leverage state-of-the-art AI approaches to enhance their drug discovery and development efforts. At Dewpoint, Francis leads development and scaling of innovative data-led approaches to enhance the company's drug discovery and clinical translation efforts. Francis, the floor is yours.
Cool. Thank you for the introduction, Diana, and for the great intro to condensates, Agnes. So what I want to do is give a perspective from the kind of Dewpoint lens. Of course, at Dewpoint, we're interested in tackling diseases through a condensate lens and finding drugs that modulate condensates to tackle hard to treat diseases, and therefore, we come to address both our drug discovery and our research into condensates using data sets like CD-Code, which have been a great resource. And so I want to give two examples today, one of how we use CD-Code to support target discovery, and the second one about how we use it really to understand more about condensate modulating drugs. But before I go into that, and before I forget, I want to say a big thank you and shout out to everyone at Dewpoint, present and past, who's been involved in this work. I haven't done as good a job as Agnes highlighting everyone for their individual contributions, but everyone who has been involved knows who they are, and it's been a huge collaborative effort. So I want to say thank you up front. So to start off, what I'd like to do is talk through an example of how we use CD-Code among other data sets to look for novel condensate targets. And I'd like to start off by saying a few words about what a condensate target means to us at Dewpoint. So we think a little bit differently about how we define a target at the beginning of a drug discovery program compared to other companies who might have a more kind of traditional or canonical definition of a target, where they are trying to find a specific protein which maybe has a deep pocket, which can be well bound by an individual small molecule or nanobody or something like that. And really what we're thinking about in terms of a condensate target at Dewpoint is a condensate whose phenotype reads out a disease state. And what we mean is a condensate where the phenotype is different between the healthy condition and the disease condition we're interested in a way where that phenotype tells us about the underlying dysfunction of the disease. The point being, therefore, that we can use these condensate readouts to find small molecule drugs that can reverse the disease state back towards the healthy state. And so what we do a huge amount of work on is using high content confocal imaging of condensate phenotypes together with associated assays like functional assays or counter screens to try and find small molecules that can revert the disease state. And that is quite differentiating because that can help us find novel condensate modulator mechanisms where we don't have to define a priori the specific target that we want to bind to. We're more looking for small molecules that actually have a network effect in reversing the condensate dysfunction of the disease back to its healthy state. We also, of course, come to our discovery programs and to our target discovery efforts from the condensate lens in terms of the data that we integrate. I'll spend most of this section of the talk talking about how CDCode fits in, but of course it represents condensate-specific data and knowledge that we integrate into trying to find new targets that read out on these diseases we're interested in, alongside the more general data sets and knowledge bases that pretty much everyone else in the space uses in terms of protein-protein interaction networks, etc. And we believe this helps us, integrating knowledge like CDCode, generate hypotheses that others wouldn't come to without looking through this angle and this lens. And lastly, we also validate the condensate targets that we're interested in a different and I think important way compared to how a lot of other folks think about validating targets. So we combine high content imaging. So for example, immunofluorescent staining of specific condensates with antibodies with omics to look for these differences between a healthy and a disease state, which we can use as a readout. And that's important because, for example, if we just look at transcriptomics or we just look at general morphological changes, for example, with cell painting, we might miss differences in the condensate, which is key to the disease, but maybe is not picked up in transcriptomics because it's a change in the tertiary structure of the involved proteins or the condensate, or maybe a change in its localization. So basically, we look at targets in a different way. We bring different data sets and a condensate-specific lens into how we look for them, and then we actually validate them in a sort of condensate-appropriate way. And this is a sort of 30,000-foot view of what makes our thinking about target discovery unique at Dewpoint. So what does it actually look like in practice? Well, this is a sort of schematic representation. I'm not sure. We've applied this similar approach across many programs with great success in multiple disease areas now. It always looks slightly different depending on the disease biology, but this slide gives a sort of high-level representation of the broad brushstrokes. And broadly, what we do is we combine omics knowledge with other data sets and integrate them together through knowledge graphs into networks, which we can then apply algorithms to and machine learning to prioritize potential condensate hypotheses. So just to step through this in a little bit more detail, in the first step, we start with gene disease or genes, which are nodes which seed our knowledge graph network, and we identify these genes based on omics associations with the disease of interest. So that might be a GWAS study or some transcriptomics or phase-specific proteomics, for example, where we're looking for genes who have a statistical association with the disease. And what we then do is use other data sets to expand those disease-associated genes into a network of multiple different node types which represent the biology underlying the disease. And this is where we bring in the condensate information from CDCode and other sources, and that's where I'll talk about more in the next few slides. We also then make predictions about which of the genes are likely to form condensates or to phase separate using models like PICNIC, which Agnes talked about. And we combine those predictions with network algorithms to basically look for nodes that we call integrators, so that are sort of well-connected nodes at the heart of the disease biology and the disease network, which are likely to form condensates, which we think are potential sort of regulatory hubs, whereby if we can modulate those hubs, those condensates, we have a good chance of redressing the disease state back to the healthy state. And so from these algorithms, we then nominate shortlists or long lists, depending on the capacity of our wet lab colleagues, for them to go and validate using imaging, for example, as I said, immunofluorescent imaging, or omics, and also using things like tool compounds or gene perturbations to validate that modulating that condensate actually will have the effect we're interested in. What we then do is prioritize the ones we're most interested in, and we go and use these as the starting point of drug discovery campaigns. So they go into high throughput screening, where we look for novel small molecules, novel chemical matter that will modulate the condensate target of interest and could act as potential drugs for those diseases. So I don't have time to go into all of those steps in detail, but I want to talk a little bit more about building of these disease-specific knowledge graphs and where CD code fits in. So this slide just talks a little bit about how we go from, on the left, individual genes or sets of genes that are associated with the disease of interest through to the kind of graph model of the disease biology. So as I say, these disease-associated genes usually come from sort of population level omic studies. So for example, GWAS or transcriptomic studies. And sometimes we add on algorithms that look at the regulatory networks or predict how these genes interact with each other. We then use these as a starting point to expand step by step into this model. So we start by adding in other connections from literature and public data sets that we know each of the disease-associated genes have. So for example, what other genes or what other proteins do they interact with? Do they interact with RNA? Are they expressed in specific tissues? Do we know that there are certain drugs that interact with these, etc.? And then we also build in the condensate information, and that's where CD code comes in, where we add in for each of the genes information of the type Agnes illustrated as it contained within CD code. For example, does the protein or the gene phase separate into a known canonical condensate? Is that condensate associated with the disease? And are there any CMOD tool compound drugs out there which interact with those condensates? And by combining all this information together, we also, as I say, use these machine learning models to predict which of the genes are likely to phase separate. And so building these steps together, we end up with this network representation of the disease of interest. And so it's effectively trying to capture in silico a model of all the relevant interconnectivity and pathways that underlie the dysfunction of the disease so that we can try and look for hubs to go and drug and condensates to go and modulate. And that was probably a bit of a kind of abstract, hand-wavy representation of what we're trying to do. So I wanted to add this slide to make it a little bit more concrete. So this slide is just an example kind of small slice or section of one of these knowledge graphs that I'm talking about. And this is focused on a graph that some colleagues built for dilated cardiomyopathy, so a cardiovascular disorder, genetically underpinned. And it has at the center a condensate of interest called BAG3, or a gene called BAG3 that forms a condensate. And you can see on the right here the sort of simplified schema that was baked into this knowledge graph, where we have genes that can interact with each other, that can be part of condensates, are known to participate in biological pathways, can be targeted by compounds, and are associated with diseases. And we build in to the node size sort of representation of how likely each gene is to form a condensate. And so these networks, you can see this is just a small slice of the network of nodes that are one hop or one connection away from BAG3. But you can see they very quickly get very complicated and hard to follow visually, if I can say that. So that's why we start using algorithms to analyze them and try and find out, what are the sort of algorithmic hubs at the center of these networks. So hopefully that gives you a bit of an idea of where CD code fits in. And I just want to end this section with one case study of how powerful this approach can be and how important it how well it can underlie drug discovery programs. So this is an example of work from many colleagues that we have been doing over the last few years in collaboration with Novo Nordisk, where we've been particularly interested in type 2 diabetes and insulin resistance. And we used a very similar approach to validate more than 60 novel condensate targets. So we started predominantly with genes associated with type 2 diabetes from GWAS analyses of UK Biobank. And we then, from those genes, built a graph representation of the disease biology of type 2 diabetes, including condensate information. We used the algorithms that I mentioned to then nominate 200 condensates, which we predicted to phase separate, or predicted condensates, I should say, which also were sort of regulatory integrator hubs within the network. And those were then tested in both healthy states and disease states by colleagues with high content imaging. And you can see some examples at the bottom of specific condensates that had a statistically significant difference in their phenotype between the healthy state and the disease state. So for example, in terms of the shape or the intensity or even the localization of the condensate, that we could potentially use as a phenotype for a high throughput screening campaign. And a subset of those were then further validated with things like functional assays or perturbations and tool compounds. We prioritized 5, and we've since been following up 2 into full high throughput screening programs, and we have some interesting, early candidate drugs coming out of those programs, which is very exciting. So that kind of wraps up a whistle-stop tour example of how we feed CD code into some of our drug discovery efforts. And in the interest of time, I'll move on to the kind of second example before I can jump and take questions at the end. So the second example is sort of less directly feeding into our drug discovery efforts, but relates more to how do we do, I guess, sort of more basic research into condensates and condensate modulating drugs, CMODs, through CD code. So one way that we've used CD code is combining it with some internal data from an experiment called Dewpoint to ask questions about how the physicochemical properties of CMODs differ from non-CMODs. So from CD code, we took the CMODs that have been curated in the second iteration of CD code, and we filtered it down to small molecule CMODs that pass some filters, like they're not flagged by Payne's filters, and they're less than a kilodalton. So we have about 250 of those. We combined it from data from Dewpoint, which is an internal assay developed by colleagues of mine at Dewpoint, whereby they ran high throughput, high content imaging of tens of condensates in parallel. So in this specific data set, it was about 12 condensates with 14 different antibody-based markers. And you can see some of the example condensates. There are canonical condensates like stress granules and the nucleolus, for example. And they ran around 4,000 compounds from a diversity library of small molecules. So these are all compounds. Some of them are approved drugs. Some of them have been tested in clinical trials but were not approved, or some of them are sort of drug-like preclinical compounds. And colleagues ran analyses to basically split this library into CMODs, which were compounds which modulated the phenotype of at least one condensate in the panel, and non-CMODs, which had no effect on any of the condensates. And of course, it's a little bit of a flawed definition, because some of these non-CMODs might be CMODs in other contexts. For example, they might modulate other condensates that we haven't looked at, or they might modulate the same condensates in different cellular contexts, for example. So this was just in a single cell line. But regardless of some of the limitations, this gives us a CMOD and a non-CMOD set of data, or multiple sets of data, where we can start asking questions like, are there chemical distinction or physicochemical properties that differentiate CMODs and non-CMODs? How do they differ? Do we see differences across CMODs with different mechanisms, et cetera, et cetera? And so these questions are some of the sort of examples that I want to spend the next few minutes just giving you some kind of illustrative answers to, of the kind of, I think, interesting insights we find. So on this slide, which is a little bit dense, but hopefully I can walk through it without too much confusion, is looking at the different physicochemical properties between CMODs and non-CMODs, whereby the analysis that we've done on both data sets suggests that CMODs have a distinctive sort of fingerprint of chemical properties compared to non-CMODs. And in particular, the CMODs are characterized by higher valency, by the presence of more rings, particularly more aromatic rings. They will tend to be less soluble, and they're often larger in terms of molecular weight than non-CMODs. So the left-hand side is 16 different box and whisker plots. They're probably a bit hard to see, but they represent different chemical descriptors with the CMODs in kind of this pinkish color and the non-CMODs in blue. And the purple stars are labeling the 13 out of the 16 of these properties where we see a statistically significant difference between the CMODs and the non-CMODs. And you can't see from these data, but you know, when you look into the actual metrics and the tests, the properties that have the greatest difference are these ones I mentioned to do with valency, aromaticity, solubility, etc. And you know, I think this makes a lot of sense given you know what we know and what Agnes described about condensates. You know that they're these dynamic assemblies of biomolecules held together by often many weak interactions between disordered regions of the constituent biomolecules. And therefore, it seems intuitive that CMODs or small molecules that interact with condensates, that is the CMODs, have these properties that seemingly would make them maybe more able to interact with the many weak interactions that govern the condensates. So, for example, that they're larger potentially gives them a greater sort of surface area to interact with more weak interactions. Higher valency and more aromatic rings potentially enables more interaction with the kind of weak electrostatic forces that govern the condensates. We also know that condensates often have a sort of distinct and often more hydrophobic solvation environment internally. So maybe the hydrophobicity of the CMODs helps them partition into the condensate. So I think what's really interesting is that even with these kind of relatively simplistic and flawed data where there's a lot of caveats, we can see this quite striking difference between the CMODs and the non-CMODs. And I think what's important for us is that, you know, these kinds of findings are both interesting, but they also have implications for how we think about drug discovery. So, for example, if you're thinking about designing a library for a screen to try and find CMODs, you might look towards biasing the compounds towards having these kinds of properties. Or if you're trying to optimize CMODs with a lead optimization campaign, you might look to change the heuristics from the sort of those that are used in traditional medicinal chemistry towards maybe some of the ways that CMODs differ. I won't dwell on this slide too much, but this is just to say that we also analyzed the sort of similar data from a machine learning perspective rather than a traditional kind of statistical perspective. So we built a machine learning model or many iterations of a similar model in a tenfold cross-validation setup to predict whether a given whether a given small molecule was a CMOD or not. And you can see on the left-hand side in the dark gray, the performance of the model across validation folds, that is on basically compounds the model had not seen during training. And the model was the training set and the testing set was balanced. So there was the same number of CMODs and non-CMODs. So chance performance is basically 0.5 on all of these metrics. And you can see that the model does, across the board, kind of statistically significantly better than chance. So it's basically further evidence that indeed there does seem to be some kind of fingerprint which the model can learn to distinguish the CMODs and the non-CMODs better than you would expect if no such fingerprint existed. And when we look at the features that the model used most to guide the prediction, again, it's a similar kind of set of things that were highlighted by the traditional metrics. So things like valency, solubility, molecular weight, etc. So kind of intrigued by this fingerprint, we wanted to sort of also see what other kind of questions we could ask about CD code. And so a natural place that we looked next was into mechanisms and the mechanisms of action of the drugs. So CD coding includes information about the targets of the small molecules. And a lot of them are quite granular, and therefore it's, you don't have that many of different drugs for each target. So it was hard to sort of quantitatively analyze the very granular annotations. But what we did was aggregate them up into kinase and non-kinase inhibitor categories. And when we did this for the CD code dataset, and actually the same for our Dewpoint dataset, we see that the CMODs were enriched in kinase inhibitors. So you can see on the left, about 34% of the compounds in CD code that that once we applied the filtering, about 34% of them were kinase inhibitors, compared to about 12% in the non-CMOD data set, which is a statistically significant enrichment. And again, it's something that makes kind of intuitive sense given what we know about condensates and kinases and kinase inhibitors. So of course, phosphorylation adds a negative charge and so changes the electrostatics and the valency of the condensate that is phosphorylated, or the target that's phosphorylated. We also know that IDRs often have regions that are heavily targeted by kinases and heavily phosphorylated. So again, it makes intuitive sense that CMODs might be commonly or enriched in or might frequently modulate kinases, which then either directly or upstream affect the properties of the condensate and therefore change its behavior. Just to say again, there's always a few kind of caveats with these things. I think it's important to bear in mind that, as mentioned, the CD code and also the Dewpoint data is limited to a single cellular context, I think, as a couple of folks have pointed out. And therefore, there are potential biases we need to keep in mind, like kinases maybe are relatively well expressed across different cell contexts and model systems, maybe more than some other targets, and maybe they're therefore more identifiable as targets than other targets. But I think with those caveats in mind, the degree of the enrichment is pretty striking. And maybe just one last result before I wrap up, which I think is particularly important and promising for us at Dewpoint and those of us trying to find therapeutically promising CMOD drugs, is the various evidence that we see that CMODs can be specific to individual condensates and to have structural characteristics or structure-activity relationships that map them to specific condensates. So what we did here in the top left is take the CD code CMODs and convert their structure into a vector representation, so a kind of embedding or fingerprint using an AI model, and then cluster the CMODs just based on their structure. So this was a hierarchical clustering algorithm which gave us 20 different clusters of CMODs just based on the CMOD structure. And what we then did was basically check whether any of the clusters were enriched in CMODs of specific condensates. And what you can see, for example, in this cluster 3 up here, is that you can see a cluster of CMODs with distinct structural properties that are enriched in CMODs for a condensate called inava. And we see similar kind of examples within the Dewpoint data for different condensates. And so this is, I think, an important indication that, or part of the expanding evidence, I don't have time to go into all of that, CMODs can be selective or specific to individual condensates, but also that they can have structure activity relationships that map them to a given condensate. And that might reflect that some condensates are preferentially modulated by specific mechanisms of action upstream, or it could reflect that they have certain biophysical or physicochemical properties that favors interaction with CMODs of a given structure and properties. I think either way, what's important for us looking for therapies is it suggests that we can learn, whether in silico or just based on experimentation, the structure activity relationships that govern this selectivity, and therefore we can bake in selectivity to our drug discovery campaigns to try and build therapies that are more selective and therefore have fewer off-target effects over time. So I won't go into this, but just this, I think I've just alluded to it as I've gone, that I haven't shown the internal Dewpoint results, but what's remarkable is that the Dewpoint data and the CD code data are very closely aligned. So we see again a very similar physicochemical fingerprint for CMODs for CD code and for Dewpoint. We see similar kinds of enrichment, for example, as I say. So just to wrap up then, it's probably been a bit of a whistle-stop tour, but happy to take questions. I think what I've hopefully done, or at least tried to do, was to give you two examples that give you a bit of flavor of how we use CD code at Dewpoint. Firstly, in terms of as an input along with other data sets into some of our primary drug discovery efforts, for example, for condensate target discovery. And secondly, as a resource or a tool for better understanding condensates and CMODs that then downstream give us insights into their potential as therapies and hopefully help us optimize how we discover CMODs in future. So with that, I'll wrap up and happy to take questions.
Great. Thank you so much, Francis, for this great talk. We have a question from Rishav. Would you like to unmute and ask your question?
Hi, thank you for the great talk. I was just wondering if you can tell us a bit more about Dewpoint. I was very intrigued by the independent sort of validation, and so wanted to know a little bit more.
Yeah, I can tell you a little bit more. I'm definitely not the originator of this. So there's others at Dewpoint who I'm sure you could get in touch with who can tell you much more. But yeah, I mean, in short, this is a sort of imaging assay which stains for multiple condensates in parallel through antibody markers. And so these are not stains necessarily in a single well. So for example, we might multiplex 4 or 5 condensate markers per well, but then across multiple wells, we can effectively stain for tens of condensates in parallel. And given the high throughput nature of it, we can still then screen, thousands of potential small molecules or other kinds of perturbations like, gene knockouts, for example. And so we use this assay in a number of ways. At Dewpoint, one example is to look at selectivity or specificity. So, for example, if we found a drug that we're interested in for one of our drug discovery campaigns, we might run it through the Dewpoint assay to check whether it interacts with any other condensates or how it does so, to help us understand selectivity.
Thank you.
John can add some more.
Hi, everyone. Just to add one extra way we use Dewpoint to what Francis just said. So kind of similar to how people have used cell painting to try to predict MOAs, where you take known MOA compounds and see if your compound of interest clusters with them. We can basically do the same thing, just using all the condensate markers instead of the cell morphology markers. So that's another kind of impactful way we've used it here. Thanks.
Thank you, John. We have another question online from Lila. Would you like to unmute, please?
Sure, thank you. So I was wondering if you have used TCGA data integration into structural analysis of condensates and generating UMAPs, considering that there are some hotspots in target proteins.
Yeah, that's a good question. So the UMAPs that I was showing and the analysis that I was showing was based on the structure only of the CMODs, the small molecules, and not of the condensates themselves. So actually, that was just looking at the small molecule structure and not the condensate structure. But I think your point's a very good one. There's a lot more that can be done with this, and relating the small molecule structure, the CMOD structure, to the condensate structure in both, with and without mutations, I think is a very good suggestion.
Thanks.
Any other questions for either of the speakers? If not, let's give one more round of applause to our speakers, Agnes and Francis. Thank you very much for really exciting talks. And thank you, all of you in the audience, in the room, and online. And I would also like to give a shout out to the team here who makes the KTT possible behind the scenes. So Jennifer Tallman, who's supporting with editing and marketing, and Curtis Bosky for IT support. Lastly, the recording will be posted on condensates.com and YouTube within a week. And yep, take care and see you all at the next event. Bye.